mountain range in the world with the Himalayas and Mount Everest. But moved at a young age to America and eventually settled in Seattle, where he grew up and also pursued his education as an undergrad. And you can see at the bottom left the Rainier Vista, which is probably visible about 5% of the time on the campus there, but the snow-capped mountain theme remains in persistence. And he got an introduction to research there, working on projects related to single force microscopy at really an atomic level to create a molecular tweezers to study like pili and appendages on E. coli, and was recruited to a number of graduate programs. But I remember in our admission system here, very highly regarded by all who met him, both professors and students, and became a student in the biomedical sciences graduate program. And in talking to Saugat early on, it was clear that he was interested in not only studying microbiology and questions related to medically important issues like antibiotic resistance, but in bringing a systems biology analysis to that. And that allowed us to forge and ultimately construct this unique dual mentorship perspective, where principally Saugat performed computational biology studies in the systems biology research group, led by Dr. Paulson, but focused on issues that collaborated with our laboratory on leading medically important human pathogens like Staphylococcus aureus and the emerging drug resistance and virulence mechanisms of those pathogens. And I can just say, as a co-mentor in this situation, it was a lot of fun all the time we spent with Saugat. He did quite a number of bench investigations, including a rotation project that was published with Nina Gao, another graduate student who defended last year. Worked closely in our lab with a former postdoctoral fellow, Nick Dillon, who you see there, who's now a professor at the University of Texas, Dallas, and also mentored other students in the laboratory, including Natasha Menon, who's a PhD student and actually a postdoctoral fellow, Yujiro Hirose, on learning some of the techniques that you'll hear about in independent component analysis of bacterial transcriptional regulation. And this power team here of former Paulson Lab and Systems Biology Research Group members, John Monk and Anand Sastry, together with Saugat really being the bridge between the two labs, generated a tremendous amount of research. Saugat also got on the highly competitive UCSD genetics training program grant and was able to present his research a number of times in that forum. And I would just say that, Saugat has really been prolific in the laboratory. He has already published 2 first author papers, but we have 2 other completed first author papers whose Google Docs are just being tightened up and will be submitted, I think, with ambition of getting published in some really nice venues. There have been 3 additional co-authored papers that were joint between the Paulson and Nizet lab. He had 1 additional co-authored paper just with my lab, but an amazing 10 additional co-authored papers with the Paulson lab. So that's a prolific output for a PhD student. And I think I'm going to turn it over now to Bernard to tell you a little bit about his perspective of Saugat in the Systems Biology Research Group, and maybe give you a hint of the next step where Saugat is going to, because Saugat has been a major contributor to collaborative research that we did as part of a systems biology of antibiotic resistance grant. And the work that he's generated really inspired a resubmission of another even more ambitious program grant that we're really proud of, and keeping our fingers crossed that we'll keep the tradition of his work going on in the lab. So Bernard, do you want to say a few more comments and then turn it over to Saugat?
Yes, I would. Thanks for that, Victor. That pretty much covers Saugat's story. His big day has arrived and time to defend. I would say that UCSD was able to offer him a very rich intellectual environment by having him placed in 2 laboratories, a leading laboratory in infectious disease in a medical school, and on the other hand, a laboratory that is doing computational biology and data analytics over in the engineering school. Not too long ago, the distance between these 2 domains of expertise was like this. Saugat is one of these, is one of the members of the new generation where it's maybe going to be like this in the future. And I think the fact we could offer him this productive and creative environment is reflected in this enormous number of papers that you're going to have on your CV. And the fact that you were co-author with so many other people shows how collaborative you were and how helpful you were to others in their pursuits. So the floor is yours.
Thank you, Victor, and thank you, Dr. Paulson, for those really kind words. Let me share my screen. It's going to take a second to set up here. Let me hide that. Great. Yeah, thank you again for those really, really kind words. And thank you to everyone in the audience for attending this talk today. So I want to talk a little bit about some of the work that I did in my grad school career, and that's been summarized in my dissertation titled The Structure, Dynamics, and Evolution of Transcriptional Regulation in Staphylococcus aureus. So my work really has 2 main ingredients, so to say. The first is, of course, the bug Staphylococcus aureus. And the second,
Saugat, I think we're just seeing an announcement for your thesis. Are we supposed to be looking at slides now?
Yeah, I'm not sure why it's not. Sorry, let me try sharing again. Does that work better?
That's better.
Okay. And let me hide that. Okay. So really, it has 2 ingredients. The first is the bug, Staphylococcus aureus. And particularly, we're going to be focusing on this methicillin-resistant version called MRSA for short. And the second part of it is going to be this technique that we're going to be using to understand the transcriptional regulation in Staph aureus. And the technique is called independent component analysis. So my introduction is also split into 2 parts. So let's get started with that, starting with the bug, Staph aureus. Now, Staph aureus causes a huge clinical burden, even here in the United States. Pre-pandemic numbers in 2017, it was estimated to cause almost 300,000 hospitalization and almost 10,000 deaths every year. And because of this large number of infections and growing number of antibiotic resistance that's popping up in this organism, the CDC has labeled it a serious threat pathogen. Now, if you sort of zoom out and look at it, all the infections throughout the world, there are many different lineages that of staph that infect and are prevalent in different parts of the world. For our purposes today, we're mostly going to be focusing on this one lineage called ST8, sequence type 8. And ST8 is prevalent mostly in North and South America. And even within this lineage, ST8, there's a MRSA lineage called USA 300 lineage. And this is a community-associated MRSA, meaning that this particular strain has sort of escaped the hospital and it can now spread really rapidly in the community, adding to its clinical burden. So that really is going to be the focus of our work here today. Staph and USA 300 is of course, a very versatile pathogen. It can cause a lot of different types of infection, but the most common being skin and soft tissue infection, so the SSTI. And incredibly enough, it actually can also colonize our nasal passages, so you can just survive there without causing any infection. And USA 300 in particular is really good at surviving in the environment outside the host. And that has, it's thought to be part of the reasons why it's able to spread so quickly in the community. Now, if you can imagine, these are all very, very different environments, and staph is able to adapt to all of these different environments. And part of the reason it's able to do that is because of its transcriptional regulatory network. The transcriptional regulatory network, all it does is it takes the inputs and signals and cues from the environment, and it then shapes the expression profile, the gene expression profile of the cell, so that the cell is better able to adapt to that particular environment. So it's a set of transcription factors and other regulators that shapes this expression profile. Now, in the past, when people have studied the TRN, the transcriptional regulatory network, the output is usually, something, a figure like this, where it's a static image that shows, arrows of some regulator controlling the expression of some genes and so on. But the issue with, figures like this is you can't compute with these static images. You can't do any calculations with it. So we wanted to make a model of the TRN that we can compute and calculate things with. And in order to do that, we turn to this technique called independent component analysis, or ICA. Now, the best way to understand ICA is to understand what the problem ICA is trying to solve is. And that problem has been really nicely formulated in what's called a cocktail party problem. So the cocktail party problem goes, imagine yourself sitting in a cocktail party and you hear a bunch of people speaking. Now, our brains can really pick out one voice from another, differentiate them, tell them apart. But how can we teach computers to do the same thing? And so the solution to that is ICA. But in order to do that, first we have to collect the data. So in order for us to be able to differentiate these different voices, these different what's called source signals, all we have to do is first place microphones around the room. And each microphone is going to pick up some combination of these voices, depending on its placement around the room. And once we have all that data from all the microphones, we put that all together and apply ICA to it. And the output's going to be individual voices from each people. And actually, you can also get the volume of each of these individuals. So we get the signal and its activity. So we can think of the TRN in an analogous way. Instead of people, you have transcription factors, so transcription factors 1, 2, or sigma factors, all these regulators. And each one of these regulators are sending out some signal that's controlling the profile, the expression profile. And so all these signals crisscross. And instead of putting microphones around the room, now we profile the expression level of all these genes in all these different conditions. We gather all that data, apply ICA, and similarly, we should be able to reconstruct the signal from each of these transcription factors. And analogous to reconstructing the volume, we should be able to reconstruct the activity of these transcription factors. So intuitively, kind of making that analogy, it makes sense why we would use ICA, but to make it more concrete, there was actually a benchmarking paper that came out in 2018 that looked at how different methods of reconstructing TRN fare with known modules in E. coli and yeast, these model organisms. And it turned out that ICA is actually one of the best methods to reconstruct the modules. So it added an additional concrete reason for us to go with this framework. And in addition to this, as we go through this presentation, we will see that ICA, the model that comes out of ICA is also going to be scalable. So as we add more and more of these expression profiles, this RNA sequencing data, we're going to get more coverage. And the fact that we're going to be able to keep up with this exponentially growing data deluge and put that into our model. Second is going to be interpretable. So the model is going to be a white box model, meaning we're going to be able to look into the model and see what it's doing. And that's going to be really, really important in understanding the underlying biology that we're modeling. And last is going to be a functional model. So as we'll see, we can combine the ICA models with other modeling techniques, such as modeling of metabolism. And then this combination is going to help us analyze different complex biological systems. And with that, we get to sort of the meat of the presentations, and that's split into three parts, into three aims. In the first part, we're going to be looking at the structure of the TRN. We're going to look deeply into what it takes to build this model of TRN using ICA and what can we learn from that structure. Second, we're going to be looking at the interaction of TRN and the metabolism, trying to understand how the dynamics of the TRN shapes central metabolism in Staph aureus. And last, we're going to be looking at the evolution of TRN. So how did the TRN evolve during the emergence of this MRSA strain, USA 300? And let's start with the structure first. So previously, I sort of gave this schematic of what ICA looks like and what it outputs, but here we're going to do a deep dive into what it looks like in practice. So like I mentioned, we start out by collecting data, and for our first model, we collected about 108 samples. So this is 108 RNA sequencing data in all the different conditions, and we collected it into what we call precise. And once we have this data, we can then apply ICA to it, and it's going to give us two outputs. So the first output is going to be what we call signals matrix, and this matrix is going to tell us what genes are co-expressed. And the second output is going to be this activities matrix, which tells us when are they expressed. And so let's look into the signals matrix. So this signals matrix has, in this model, 29 components. And so we can look into one of these components to see what's in there. So here on the right, I have mapped out one of the components. And in this component, you have waiting for all the genes in the Staph aureus genome. And as you can see, most of these genes have zero weightings or near zero weightings. So there are these gray bars here. But some genes are these outliers that have this high weightings. And if you closely look at the gene names, they all have this, almost all of them have this prefix PYR. So this is really easy to recognize if you work with Staph or any other microbes. This is pyrimidine biosynthesis genes. So all these genes that are the outliers here are actually regulated by PRR regulator. And so we can really easily tell that this specific component is a signal from the regulator PRR. And the genes here that are outliers, we call imodulon, which is short for independently modulated sets of genes. And so in addition to getting what's in this imodulon, we also get its activity from the other matrix. So if you look at the corresponding component in our activities matrix, we see the activity of this PRR regulator across all 108 samples up here. And we also can take a look at, if you're interested in any specific media, you could also take a look at those media and do hypothesis testing to see if that regulator activity is different in those specific conditions that you're interested in. And so this is just one component that's associated with this one regulator, but we extracted 29 total components. And if we map all those components, we're able to map almost all of them to specific regulators within Staph aureus. So these are metabolic regulators, different sigma factor and stress associated regulators, and so on. But we also get some interesting imodulons that we can't quite pin it to a specific regulator, and these we call functional imodulons. And a great example of that is this phi SA3 imodulon. So let's take a look at that. So phi SA3 is a prophage that has integrated itself into Staph aureus genome. And if you look at the constituents of this imodulon mapped here, all the genes in teal here are present in that imodulon. And all these genes are, of course, the phage proteins from the prophase that has integrated itself. So all these genes are being co-expressed in all our samples, and that's why it's picked up as an imodulon. And we add it here as a functional imodulon. So for a lot of these, we were quickly able to recognize what the imodulons, what the signals were. But what happens if you're not able to figure out where the signal is coming from? And that was the case for one of these imodulons. So when we get data out from ICA, this is basically what we get. We get the gene weightings, and we can map it onto the genome so we can see where the gene starts. Now, if you have a imodulon like this and you're not able to map it to a particular regulator like we did with PRR, then the fact that the imodulon is a white box model comes in handy. So after looking at these genes for a while, I had guessed that this was associated with alternate sigma factor, SIG-S. And so to confirm this might be a SIG-S imodulon, I went to the literature and looked at what we know about SIG-S. And it turns out we don't know too much about SIG-S, and we don't actually even know what the regulon of SIG-S looks like, but we're still able to confirm some things. So based on literature search, we know that SIG-S, a lot of these sigma factor are auto-regulated. So that means if this is a SIG-S imodulon, we should expect the SIG-S gene itself to be in it. And we do find that there. We know that for sigma factors, that the regulation is concentration dependent, meaning that if the concentration of sigma factor in the cell goes up, that means its regulon's expression level also goes up. And so we see that here as the SIG-S expression level mapped out in x-axis here goes up, the activity of this imodulon also goes up. Through some studies, we also know that this cysteine responsive regulator SIM-R represses the activity of this sigma factor. And so we can also map that out here as the SIM-R imodulon activity goes up, SIG-S activity goes down. We know that these alternate sigma factors have specific motifs that they bind to. And so we should be able to find that in this imodulon too. And so all we did is gathered all the genes in this imodulon and looked for conserved motifs upstream of the genes. And once we found them, once we found this conserved motif, we compared it to all the other known conserved motifs in bacteria, and our closest match was alternate sigma factor from Bacillus subtilis. So it seems like that alternate sigma factor motif is also present. And lastly, it's part of this, what we dubbed fear versus greed response. And this response is something that we see again and again in a lot of different bacteria, and even in one of these archaeas that we have built these imodulon models for. And it's this trade-off between this translation imodulon, which contains all the translation ribosomal genes and other translation-associated genes like initiation factors, and all these stress-associated alternate sigma factors. Their activity always seems to be negatively correlated. And if we think this is a SIG-S imodulon, we should expect the negative correlation, and we do see that here. And so we can gather up all these different evidence and for the first time propose a regulon for this alternate sigma factor. And what I really like about this example is that we didn't necessarily have any single sample in our input data where we try to isolate signal from SIG-S. It's just as we were collecting all these RNA sequencing data, the SIG-S activity was changing in the background, and ICA was able to pick it up. And because we were able to look into the model, we were able to then label it as a signal associated with this sigma factor. And so now that we have the structure, there are a lot of really cool science we can do with it. But I wanted to kind of focus on this one utility because I think a lot of biologists here will find this useful. And this has to do with this differential expression level. So here, this is a plot called a volcano plot. And all it does is it tracks differential expression level of different genes in conditions. So here we're looking at these two media, CAMHP versus RPMI. So each dot here represents a gene. On the x-axis, we have log-fold change in expression of that gene between these two media, and on the y-axis, we have the p-values. And the colored dots are the ones that are significantly differentially expressed. Now, this is a very typical plot that you're probably used to seeing if you've worked in biology. And here we have over 850 genes that are differentially expressed, and they're from all the different COG categories. So they're colored by the COG categories. And it becomes really, really difficult to interpret what's exactly going on. What are the genes and what are the systems that are being changed when we change these media conditions? But if we instead look at this exact same data through imodulon activity instead, we get a much more interpretable data. And this is what we get. So it's the exact same data. And instead of looking at individual genes, we can look at the change in activity of regulators. So we know our, for example, PRR, this is a purine biosynthesis regulator. So it's generating more or synthesizing more purines. MNTR is responsive to manganese concentration. So it's star for manganese and so on. So we can very, very quickly interpret our differential expression data, even if it's complex and contains hundreds of differentially expressed genes. And of course, this becomes even more important if you have time series data, because then you just have these volcano plots stacked on top of each other on this time axis. And we can convert that into our imodulon activity. You could take your favorite regulators that you want to take a look at, and you could track their activity across the sample, across the time. And so the data I've presented so far is part of this older model that we published in 2020. And ever since then, this publicly available data has been growing exponentially as they publish their papers, submit their RNA sequencing data online. We can now take that data and incorporate it into our model and make our model better by having higher coverage and more imodulons. So from here on out, I'm going to be using this expanded model for the rest of the aims. So this expanded models was generated with 386 samples versus the 108 in the original one. And it contains 76 imodulons versus the 29 in the original one. And so that brings us to the end of the part one, where we generated this first version of the model that contained 29 imodulons. And we used that model to figure out regulons for this alternate sigma factor, sig-S, that was not well understood. And we also demonstrated the utility of these imodulons in interpreting complex RNA sequencing data. And lastly, we showed that our model is scalable, and as new data becomes available, we can keep adding on to it and get better and better coverage of the TRN and Staph aureus. With that, we're going to move on to aim 2, where we're going to be looking at the interaction between the TRN and metabolism. So this project really started with this particular observation. Here we're looking at explained variants of all the different imodulons in this expanded model. And explained variants, really an intuitive way to understand it is which imodulons in the model explain most of the changes that we see in our RNA sequencing data. And so if you look at it, the three explained imodulons, highest explained variants are associated with CCPA, the translation imodulon, and the code Y imodulon. Now, CCPA is a catabolite repressor protein in firmicutes, so meaning it in presence of glucose, it suppresses expression of all the other alternate carbon sources. And it also controls lower glycolysis, TCA, and so on. Code Y can be thought of simply as global regulator amino acid biosynthesis. Lastly, translation contains 54 ribosome-associated genes. So what really got me interested in these three imodulons was reading Terry Waugh's paper. And Terry Waugh has proposed these really nice coarse-grained model of different way of sort of slicing the complex systems in cells. And one of the coarse-grained models he proposed is this model of controlling protein synthesis. So you could think of protein synthesis control in 3 steps. So you have your carbon source coming in. That carbon source is turned into these what's called intersection metabolites, usually some kind of keto acid. And that's then converted into amino acids. And that amino acid in 3rd step is, of course, polymerized into these peptides and proteins. And so what's really nice about these very simple coarse-grained model is that we can very easily try to figure out what are the underlying principles that governs the cells. And once we have that backbone, then we can use that scaffold to understand the complexities and caveats that you're inevitably going to run into when you're studying biology. And those 3 imodulons that had the highest explained variance, of course, fit really nicely into this coarse-grained model. CCPA controls the 1st step, code Y controls the 2nd step, and translation, of course, controls the last step. So now we want to understand how are these different regulators interacting with one another to control these protein synthesis in Staph aureus. And we'll start with the first 2 regulators, CCPA and code Y. So we started out by mapping out the activities of this 2 imodulons across our sample. So here each dot represents a specific sample. And on the x-axis, we have the CCPA imodulon activity. And on the y-axis, we have the code Y imodulon activity. And what we see is that when there is increased preferred carbon, so something like glucose added to the media, Staph starts expressing genes involved in amino acid biosynthesis, so those that are controlled by code Y. And so what's even more striking about this is that if you knock out code Y, then the samples sort of fall out of this plane. So those are the red dots up here. So really what this tells us is if we're able to manipulate CCPA activity, we can change code Y activity. Code Y is downstream, but if we change code Y activity, but we're not able to change CCPA activity. So CCPA here seems to be working downstream, or I'm sorry, upstream of code Y. And so this is just correlation on an expression level. Can we check it using, is there any difference at metabolic level too? And in order to check that, we turn to our metabolic modeling. And I don't want to go too much into detail about how these models work, but at a very simple level, we take our individual biochemical pathways and we can turn that into this matrix called stoichiometric matrix. And once we have it in this sort of matrix formation, then we can do a lot of cool modeling with it. So for example, we can impose constraints so we can make sure that all our reactions are mass balanced. If you want to simulate in different media, we can control what the uptake rates of different metabolites are and so on. And once we have those constraints in place, we can run simulations and ask questions like, what is the metabolic flux state that gives us maximum growth in a given media, for example? And so we have this model, and in particular, we have this model made specific for this USA 300 strains. This was done by our previous graduate students here in SBRG, Yara. She built this model for USA 300, and she was able to define these constraints for these different media. And so we took that model, we mapped our imodulons on top of that model, so that way we can track what these imodulons are doing in different conditions. And that we took that model and we constrained it using uptake rates from 2 different media, CDM, which is chemically defined media, and CDMG, which is the same media, just with added glucose in it. And then we simulated to maximize growth. And after simulations, we looked at the model and we calculated what the fluxes that are going through enzymes encoded by genes in code Y imodulon. So that's a little bit mouthful, so I'll just call it code Y fluxes. And as we can see, in agreement with our transcriptomic data, that when we add glucose with the preferred carbon, we get increase in code Y imodulon activity. So in metabolic model, they're also biosynthesizing amino acid. But this seems a little bit unsatisfactory to me because this model gives us it's an optimization algorithm, so it gives us an exact solution. So there is no variance to be accounted for. So to get around that, we can turn to what's called sampling. So if a PFPA solution gives us that optimal single solution, as pointed out here, what we can do is we can instead sample the whole solution space and figure out what are all the possible feasible metabolic states that a cell can take in each one of these conditions. And that's exactly what we did. We turned to sampling, again, in CDM and CDMG. And then we looked at flux through all these different amino acid biosynthetic pathways. And we see that in almost all these amino acid biosynthetic pathways under code Y control have higher flux when we add glucose. Again, in agreement with our transcriptomic data. So now, if we go back to our model, we can add one arrow to it, which is CCPA and code Y activities are coordinated by staff, and CCPA likely works upstream of code Y during this coordination. And next, we're going to move to the second part, which is figuring out what these intersection metabolites are. So what are the interfaces between CCPA and code Y? So if you look at the gene content between these two imodulons, CCPA and code Y, there are actually no genes that are shared between the two. However, if we map the imodulons onto the metabolic map, we see that they do intersect at these important metabolites. So one example here would be pyruvate. If we track pyruvate in this metabolic map, CCPA regulated genes here in blue drive it toward neurominic acid. On the other hand, code Y regulated genes in orange here drive it towards branched chain amino acid biosynthesis. So here, pyruvate, one example, but of course we can systematically go to this metabolic model and we identify 22 of these total intersections. So these intersections, metabolic intersections, really point to interfaces where these two genes regulated by these two regulators have to coordinate their expression and activity to optimize metabolism. So for example, we have another example here with threonine. If you have a CCPA regulated gene, it pushes threonine towards lactate, breakdown towards lactate. On the other hand, code Y regulated genes pushes it towards incorporation into branched chain amino acid. We also have other intersections that we have actual simulations data from our previous CDM and CDMG. And so one example of that is glutamate dehydrogenase. So glutamate dehydrogenase can reversibly interchange between alpha-ketoglutarate and glutamate. And in CDMG with glucose, where the cell starts making a lot of amino acids, glutamate dehydrogenase converts AKG into glutamate, and that glutamate then acts as an amine donor to drive all the different amino acid biosynthesis. On the other hand, if in the absence of glucose where gluconeogenesis is turned on, L-glutamate is turned to alpha-ketoglutarate, and the TCA cycle is fed into lower glycolysis and gluconeogenesis. So we can see that even though they're not sharing any genes between them, these two, the genes regulated by these two regulons, these imodulons, really need to coordinate with one another in order to shape the metabolism. And next, we're going to bring in the last part of this puzzle, which is translation. And in order to do that, we're going to have to move away from looking at the activity of the imodulon and go back to the gene content. And I mentioned that the CCPA and code Y imodulons do not share any metabolic genes, but they do share this one other genes, HPF. So on the x-axis, we have the weightings for the CCPA imodulon. So we see all these CCPA genes. And y-axis, we have the same for code Y. And HPF here is the sole member of both these imodulons. And HPF encodes a hibernation promoting factor. And all it is a small peptide that dimerizes 270S ribosome subunit and turns it into an inactive 100S ribosomal subunit. So in this way, HPF can control the translation rate in the cell. And now we can see that CCPA and code Y is acting in this model as almost feed-forward in a feed-forward mechanism whereby these metabolic regulators are also controlling translation rates. So now this is evidence from, of course, expression profile in our model. Can we confirm it that HPF is being regulated in any other way? In order to do that, we turn to the regulatory region of HPF. So HPF here is noted in this last arrow and can be transcribed from 2 different promoters, these 2 black arrows. And Ye Gao, a former grad student, had developed antibodies and was able to do ChIP-EXO for code Y, and found that there are 2 actual code Y binding sites that can influence HPF expression. And if we look at the sequence right upstream of the second code Y binding site, we see a CCPA binding motif also present. So now we have evidence that both CCPA and code Y from our transcription data and our looking at our sequence, that they're both regulating HPF. And so this architecture actually with 2 code Y binding sites with CCPA in the middle is actually, it can be found in Bacillus also, it also contains CCPA and code Y. And in this promoter here, you can see code Y binding site 1, code Y binding site 2, and right in the middle, we have our CCPA binding site. So it seems to be a preserved architecture whenever these 2 regulators are working together to regulate a gene. And lastly, we see, if we map out, going back to the activity of translation imodulon and code Y imodulon, we see they are also very closely correlated. So it makes sense that when you're expressing genes associated with amino acid biosynthesis, you're also simultaneously expressing genes associated with translation. And we see that this is actually only code Y and code Y and translation specific, that we don't see this correlation between CCPA activity and translation activity. Now we can take all this data and observations and we can start adding to our coarse grain model. So in the beginning, we saw that CCPA code Y activity are closely correlated, and we confirmed that with M models. So we can add this activity between CCPA and code Y. Using our metabolic model, we recognized what these metabolic intersections were and how fluxes were coordinated around these intersections. Third, we saw that code Y and CCPA has almost this feed-forward regulation of translation via this HPF expression. And lastly, we saw that there's a coordinated expression of code Y and translation imodulon genes. Now, at the moment, we don't know if this is a feedback mechanism or if there's another regulator that's controlling both of them, because we haven't figured out what the regulator for these translation imodulon is quite yet. So what's really nice about this modular way of building this model is we can keep adding to it. So I didn't talk about, for example, nucleotide metabolism. So pyrimidine and purine biosynthesis also closely correlated with amino acid biosynthesis in our model. So the activities are closely correlated. So in the future, we can really easily add that and keep expanding the model. On the other hand, I briefly talked about this fear versus greed trade-off earlier. And we can, of course, add that aspect into the model too, to try to understand how does stress and stress response play into this protein synthesis in staph. And so in this aim, we looked at carbon and nitrogen metabolism and how that's coordinated by CCP and code Y in staph, and how that metabolic regulation actually feed forwards into controlling translation rate in staph also. And in the end, we took all this information together from imodulons and from our metabolic models and proposed this coarse grain model of modular control of protein synthesis in Staph aureus. And that brings us to our last aim, where we'll be looking at evolution of TRN during the emergence of this USA 300 strain. So like I mentioned earlier, Staph aureus tends to be sort of regionally distributed, where you have these different sequence types, different strains that are found in different regions of the world. But if we look at any one of these genes, and if you kind of turn the clock backwards and try to look at how they emerge, we see this familiar case again and again. Usually you have some basal strain. So this is looking at emergence of MRSA in the Indian subcontinent. You have some basal strain, usually MSSA, so methicillin sensitive Staph aureus. And over time, they've gained some genes associated with antibiotic resistance. And as they gain those genes, they're able to proliferate more, and you see more and more of those. And if we look at our MRSA strain of interest, we see the same thing. There is a ST8 progenitor, and this progenitor over time has gained these genes. SCC-MEC4 is a cassette that contains the MEC-A gene, which confers antibiotic resistance, and over time has also gained other genes. So these SEEK, SEC, and LUC-SF, these are all different toxins and virulence factors that help staph survive. But if you look at all these maps, what we notice is that all these analyses only look at what's happening at gene level, right? What genes were gained and what genes were lost, and so on. And not really look at different mutations like single nucleotide polymorphism, SNPs, or indels. And the reason is that because of this sort of clonal propagation of these strains, there's a genome-wide linkage of all these different mutations as they occur. And because of this linkage disequilibrium, we're unable to figure out which genes are actually causal versus which genes are simply there because they're linked to the causal gene. And so here we thought maybe we could use our TRN model to find candidate mutations despite this linkage, because with our TRN model, we might be able to figure out how the gene regulator relationship has changed between different strains. And if those genes also, turns out if they also have some mutations that are enriched, then we'll have strong evidence to think that those are causal strains. So in order to do that, we started out with, of course, our clonal complex 8 genomes. So we collected 2,000 genomes from this Patrick database. And of course, our USA 300 model that I mentioned earlier is part of this clonal complex. And so we divided the strains into either USA 300 or non-USA 300 strains. So USA 300 here in purple and non-USA 300 here in gold. And what's really nice about working with clonal complex 8 is that in addition to having our USA 300 model that we've already built, there's also this nice reference strain NCTC8325, for which a lot of RNA sequencing data is already available online. So we're able to really quickly build a model for another strain without having to do a lot of new data collection. And so once we divide it into USA 300 and non-USA 300, then we do our classic gene association study to try to see what mutations or genes are enriched in USA 300. And we get our classic classic genes, so MEC-A, the antibiotic resistant gene, SEEK, SEC, and PVL, so those virulence factors that I mentioned earlier are enriched. But of course, we also enriched a lot of other genes and mutations that are specific, that are unique to USA 300. And now the challenge is to figure out which one of these are causal and important in clinical success of these strains. And so here we get to sort of the heart of the problem, which is genome-wide linkage. So here we plotted out all those enriched mutations. And here each dot represents a comparison between two mutations. So on the x-axis, we've mapped out the distance between the two mutations. And it goes up to 1.4 million because Staph aureus genome is 2.8 million, but it's a circular genome, so max distance you can get is 1.4. And on the y-axis, we're mapped out the linkage with this R-square value. So basically tracks how often do these mutations co-occur in the same genome. And we see that even at almost maximum distance, we get a lot of mutations that are almost perfectly linked together. And so because of this, we're precluded from figuring out which one is causal mutation. And so to get around this, some people rely on homoplazy to try to see if there are certain mutations that occur in hotspots over and over again in several independent lineages. That may indicate certain spots that are important for clinical success. And so in order to see if we can find these mutation hotspots, we then turn to Patrick again, but this time we downloaded all the genes, all the sequences for open reading frames. And to find these hotspots, we relied on this measure called Shannon entropy. As if you're familiar with Shannon entropy, here we're calculating it with where n is the number of unique alleles and p of x is probability that allele. But if you're not familiar with Shannon entropy, we can still get a very intuitive understanding of what it is. And so first we take all our protein sequences and we align them. And if at any given position, there are only one allele present, so you get perfect match all the way through, then the entropy is going to be 0. If you get some rare mutation, then you'll have a small spike, but it won't be a standout. But if you have a position where there's, let's say, a 50-50 divide between the different alleles, the distribution of different alleles, then you'll get a better spike. And at last, if you have polyallelic region, then you'll get a huge spike, noting that this is a mutation hotspot. And so we're able to do this for all our mutations in our GWAS that map to one of the open reading frames. And unfortunately, what we found is that there's actually only one mutation that occurs in a hotspot. So here we're comparing hotspot, so that's Shannon entropy measurement of a particular protein. So this is a capsule biosynthesis protein in CCH strains. So we see our hotspot here, and in non-CCH strains, so these other lineages, and we see a hotspot there again. So this seems to be an important mutation hotspot for staph during its evolution. However, this is the only exact match we got. So for all the other ones, there were spikes nearby, but they were not an exact match. So the Cap5e, the example here, this 2 spikes are right on top of each other, so 0 distance, but all the other ones were either 1 base pair off or 6 base pair off, and so on. So this sort of is almost a nightmare case scenario. You have all these enriched mutations. They're all in linkage disequilibrium, and all these mutations tend to be de novo mutations, unique mutations. So you can't even rely on homoplasy to try to figure out which one is causal. And so to resolve this issue, we have one more trick up our sleeve, and of course that is going to be with our imodulons. And so, like I mentioned earlier, it was important that we had this other reference strains, NCTC8325, that is a non-USA300 strain, because we were able to quickly take our original USA300 data, combine all the data that we got for that NCTC strain, and apply ICA to it to get this one model that is a combination of 2 different strains. And we were expecting to get a strain-specific imodulon. And strain-specific imodulon is actually really, really easy to spot. We just look at the activity of all these of the imodulon, and we label it at USA300 in blue here, or NCTC8325, the other non-USA300 reference strain. And we see that the activity of this imodulon is, as the name implies, strain-specific. USA300 has one activity level, and non-USA300 has this different activity level. Now, if we look at the constituents of this imodulon, we see all our familiar gene, MEK-A, that antibiotic resistance gene, SEEK, SEC, and LucSF, those virulence factors that we talked about. So in addition to that, of course, we also see a bunch of other genes that are conserved, like this ISDH genes. So now all we have to do is find genes where mutations were enriched in our GWAS analysis, and that are also enriched in this imodulon. And we found 3 of those genes, but I'm basically only going to focus on one, which is ISDH here, because the data here is really easy to interpret. So what is ISDH? So ISDH encodes this heme scavenger. So basically, whenever staph is starving for iron, it will secrete this protein, which will go out there and bind heme in your blood, and it will take it back in, and it can recover the iron within. And it's regulated by this repressor called FER, ferric uptake regulator. So if staph has enough iron, then it will no longer need to produce this protein, and so FER then represses its expression. Now, if you look at the regulatory sequence of this gene, we see why this regulatory region was enriched in our GWAS. We see that if we start at the translation site here and the promoter site here, see that there's a 38 base pair deletion in TCH1516, which is a reference strain for our USA300. So all our USA300 strains have this 38 base pair deletion. And what's really important about this deletion is that the FIR binding site, the transcription factor binding site, is within this deleted region. And so that would explain why our ICA analysis recovered this gene as one where there was strain-specific difference in gene regulator relationship. And of course, if we look at our raw expression data, we can see that across our 600 or so expression profiles that we use to build our model, our NCTC samples have, in general, lower expression levels, and our USA 300 samples, where that repressor binding site was deleted, have higher expression level. And so in this way, we were able to recover a potential causal mutation, even though our samples were almost all, or our mutations in our samples were all in linkage disequilibrium. So with AIM3, first we defined SNPs and SNOL indels that were associated with the emergent strains. And we unfortunately found that a lot of these SNPs and indels were in linkage disequilibrium with almost all of the mutations that were enriched, confounding our analysis and trying to figure out which one are the causal mutations leading to clinical success of USA 300 strains. But by combining that with our imodulon model, we were able to find at least one causal mutation where the regulation of this gene ISDH has altered between these two strains. And so here we mostly focus, of course, on changes in gene regulation, but you can imagine we can do similar things if we can model other aspects of cellular biology. So for example, I know AlphaFold right now is not the best at incorporating model, but I've heard John Jumper, it's in his to-do list. So hopefully in a future iteration of AlphaFold, we can imagine incorporating that data to figure out what, to find our candidate mutations. Similarly, Errol Kavas, who was a grad student here, created did a similar project where he was able to connect different SNPs with changes in metabolism and in mycobacterium. And we could also import that here. And so we can have all these different models looking at different aspects of biology. And if we can figure out if certain aspects of biology has changed and link that to a mutation, we can make great strides in trying to understand the importance of these mutations in these emerging endemic strains. And so that brings us to the end of our talk. So in our first part, we looked at what the structure of TRN looks like and how we can model it using ICA, and how this model can help us define new regulons that are previously unknown in staph. And of course, we demonstrated the utility of these imodulons in trying to understand complex expression data. In part two, we looked at the dynamics of metabolic regulation, how different regulators that control metabolism can also coordinate with one another to control protein synthesis. And in the end, we proposed a course grade model that can be really easily expanded as we get more and more data. And lastly, we looked at the evolution of TRN during the emergence of USA 300 strains and how we can really get around this limitation of GWAS analysis by modeling the TRN in Staph aureus. And with that, brings me back to probably my favorite part of any defense, which is acknowledgement. And I would like to start, of course, by thanking my two great advisors, Dr. Paulson and Victor, of course, for all the support that you guys have provided throughout my time in grad school, but also for creating this amazing collaborative open lab space where I was able to work on so many interesting projects and just had fun doing science for the last few years. I also like to thank all my other committee members who incrementally helped me make this project better and better with each one of our meetings. And I'd like to thank everybody in SBRG, but a special shout out to CJ, Anand, Errol, Yara. You guys were both my peers and my mentors, and I learned so much from you guys. And I would not be able to do any of this science without all of your valuable input. Also like to thank all my mentors in the Zay Lab, Nina, who was my mentor from day one, and of course, Nick, I guess Professor Nick Dillon now, and Samira, and of course, Hannah from Pagliano Lab. without whom all these projects, and especially AIM-1, would not have been possible. I also want to thank all my friends, old and new people. When you start grad school, people always talk about how hard it's going to be, but nobody told me how fun it's going to be and how many amazing people I'm going to meet during this project or during this journey. I guess I would give a double shout out to CJ here because he was, an amazing lab mate, amazing friend, really supportive, and could not have done this without you, buddy. Also want to say thank you to probably the best cohort in BMS. Maybe a little biased, but maybe ever. You guys are really, really amazing and made grad school really, really, really fun. And I'm going to be really sad that I'm leaving this behind. But I want to give a special shout out to Appleton boys, Aaron, Ryan, Mike, no particular order. Emily, Jack, Chris, Cedric, I can go on and on. Nadia, Angeline, thank you guys. And lastly, I want to say thank you to my family, starting out with my brother. I've been competing with him from April 15th, 1997. I think that was the first day we started competing, and we still compete. And I hate to say it, but I think he's winning right now. Found out he's running faster than me. He has a cool job where he gets to say things like it's classified. But I think when I get my PhD, in my completely unbiased opinion, I'm going to be winning again. So your move. And of course, I want to thank my new family. I did get married during grad school and gained new family and new source of love and support. And lastly, I want to thank my mom, Sandhya, and my wife, Jamie, who's been source of unconditional love and support throughout my life. Love you guys. And that brings to the end of my presentation, and I'll take any questions from anybody in the audience.
Oh, fantastic presentation. It was a big challenge to cover so many topics, but you brought the theme together, and now we're at the time in which we will look for questions from the audience. These can either be typed into the chat where you're getting a flurry of personal congratulations, but I see Michael Valdez has a question, so we'll let him get us off and running here. So Michael?
Hey, Saugat, great talk, absolutely fantastic, and congratulations. We've talked about this a lot throughout the last couple of years, but one question I don't believe I've asked you yet is whether, so like the ICA method depends a lot on adding samples that have, hopefully incremental differences in the initial kind of starting conditions for the bacteria. And as you add additional samples that cover, previously uncovered conditions, you hopefully get variations in the transcriptional response that lead you to be able to identify these imodulons. Is there any way you can weight the discovery of new imodulons based on what variables the data that you're adding is changing compared to what data you've already had? And what I mean by that is, let's say you are changing carbon levels across samples. Is there any way you can weight the learning of the recalculation of the imodulons based on the fact that what you've added is carbon? And so it should be looking for things in relation to, carbon uptake, carbon, related metabolites, et cetera, et cetera.
So at least the version that we have now is completely unsupervised learning. So we don't have any of that other extra weightings or inputs into it. But if you want to, I guess there's two things you could do about that. First, if you're interested in what systems available, you could, incorporate it into the model, and you could just look at the activity, and that will tell you everything you need to know about what's changing when you're changing the carbon level, for example. The other thing is, if you don't want to build a whole model, but you have an existing model, you can actually project your data into the model. So you don't have to rebuild it as you add in these new expression profiles. You can just ask the model, what is changing in this RNA expression profile, even though it's not part of the model yet. So you could do it, I guess, either way, and that will sort of isolate your samples if that's what you want to do.
Okay. Yeah, I guess I was asking because I'm wondering what value performing mutational experiments might add to identifying what imodulons might exist within the reference animal, like you're a little bit like what you're doing with the strain experiments. But I imagine that it's difficult to tie those things because the underlying assumptions about the organism are different. So I guess, can you comment to that? Does comparing different strains together give you any additional information on imodulons that maybe didn't exist prior to you looking at two different strains?
So yeah, I didn't get to quite present that data. So there are other ways of comparing strains here. So we could combine these, or at least the ones I presented, we combined it into a single model, but we can also create two separate models between two strains, and we can look at the weightings of these imodulons and how those weightings change depending on what strains they are, and that could tell us how that gene regulator relationship has changed. But there are a lot of other factors that can also change the weighting, so it makes it a little bit more complicated to analyze the data in that way. So we instead went towards this combination methods. But if you have sufficient data, then you can compare sort of the weightings between the two strains to figure out what's different.
Cool, thanks.
Yeah, no problem.
Okay. Next up is John, who has a question from the chat. Can you unmute yourself?
Can you hear me?
Yeah, I can hear you, John.
Okay, so a year ago, you asked me a really deep, profound question after my dissertation defense. So I'm here for my revenge, albeit with a slightly less profound question. Can you go to your slide? I think it's like number 3, where you're talking about the algorithm, the ICA. Lots of clicking.
Let me try that again.
There we go.
Can you see that?
Yeah. Okay. So, maybe it's kind of just a naive question, but when I think about the analogy problem where you have the people speaking and you want to measure their voices, the signal that you measure in some sense is just a simple superposition of each person's voice. Whereas in the case of a cell, it seems rather implausible to me that this simple superposition linearity would hold. So my question is, does the ICA algorithm require the signals to be just linear superpositions of each other?
Yeah. So so far it does seem to be hold together, and they are linear combinations, as you have mentioned. And so for a lot of the regulators we have found, we are able to extract imodulons, but there are some regulators where we're wondering if we can't get imodulons for because it can't be simply decomposed into this linear combination of stuff. So it's possible, we will hit limit. I mentioned we could scale with data, but I'm sure maybe we'll hit limit with certain regulators and certain interactions that can't be decomposed linearly.
Well, it seems to work astonishingly well then for a nonlinear system to be modeled like this. It's very impressive.
Yeah. A good linear approximation should not be underestimated, I think.
Yeah.
Yeah. Thank you, John.
Yeah. Thank you.
I'll just hop in.
Aaron, yeah, go ahead, Aaron. Sorry.
Super great, Saugat. Congrats. I was just curious, actually, on the slide you just had up there, with the ICA kind of way of deconvolving problems. Is there a limit to how far apart your different conditions can be? I mean, is it that you have to be, essentially titrating particular chemicals, particular reagents, and you're working within that, or could it be that you're comparing when we add some metabolite and then some other totally disparate metabolite and then a third one, and be able to compare back and forth between those? How disparate can those conditions be for PCA?
Yeah, so actually you could do both those experiments and still get imodulons. So when we, at least in the current stages, what we try to do is we try to make the samples as disparate as possible, because we want to get as many different imodulons as possible. And if you have very diverse samples, then you get a lot of different combinations of regulators activating in those different conditions, and you get a lot of new imodulons. But if you want to do like a focused study where you're looking at one regulator and you're looking at how its activity might change as you're titrating different metabolites or something, you could also do that. So it depends on what your end goal is. If you want to finally characterize one, then you could do your titration. If you want to get as many imodulons as possible, if you want to get a higher coverage, then you want to do these different conditions.
Cool. All right. Thank you.
Yeah.
Okay. Any other questions from the audience in general? I was wondering, Saugat, if you could share with folks what you're going to be starting as your new endeavor immediately in the next stage here.
Yeah, it's a good question, Victor. I'm actually going to be joining a startup with John Monk and CJ that they've started based on John's work during his grad school. It's called Pastor 21. And essentially the focus is going to be trying to help hospitals and other facilities track outbreaks and antibiotic resistance so they can better contain them. We're going to be using a lot of sequencing technologies in order to be doing that. So taking some time off, but yeah, getting started in May. Really excited about that.
Let's see. Well, I think, those are excellent questions. And I know that the committee has been holding their tongue and ready to go into the closed session. So let's see, how are we going to achieve this? Jamie, are you going